In this video, I will discuss the process of oogenesis, that is, the creation of the female gamete, what we would commonly call the egg. Each and every egg, or oocyte, is going to be surrounded by a series of cells called a follicle. The follicle grows and develops as the oocyte is going to pass through the phases of mitosis and meiosis. The story begins with oogonia. These are the primordial germ cells that begin to develop kind of outside the fetus during the first couple weeks of life. Ultimately, they are going to undergo the process of mitosis to make more and more of these oogonia, and by about week 4, week 5, week 6, these primordial germ cells are going to migrate into what will become the gonads. Now, each and every one of these oogonia is surrounded by a series of cells called follicular cells. These will ultimately become the follicle, which will grow and develop along with the developing quote-unquote egg. Now again, before birth, within the first couple weeks of fertilization, in fact, these oogonia replicate, they make a lot more of themselves, and they begin the process of meiosis 1. Now this is very different from what happens in a male. In a male, the germ cells replicate and then they just kind of hang out in the testes until puberty. In females, before birth, the oogonia begin the process of meiosis 1. So essentially, the oogonia start out with one copy of chromosome 2, for example, from mom, one copy of chromosome 2 from dad. They begin the process of meiosis, so they synthesize a copy of each of these. So here's a copy from mom, and here's the chromosome from mom, here's an exact copy from mom, chromosome 2 from dad, and a copy of chromosome 2 from dad. So essentially doubling the amount of chromatids within the cell. Okay. So this is the beginning of meiosis. Crossing over really hasn't happened yet. You don't get this mixing of genes just yet. Essentially, the DNA has been synthesized, and this cell is going to stop meiosis in prophase 1. Again, this is super different from males in that once males start meiosis, they complete the meiotic process. Here, before birth, in the developing female embryo and then fetus, in her ovaries, oogonia begin the process of meiosis 1. They arrest this process in prophase 1, and we call this cell a primary oocyte. Right? Just like a cell going through meiosis 1 in a male would be a spermatocyte, a primary spermatocyte, here we have primary oocyte. Okay. At this time, the follicular cells haven't really changed. It's pretty much just a single layer of flat or squamous cells all the way around this spherical oocyte. Within the ovary, these primary oocytes surrounded by the primordial follicle are condensed into what are called egg nests. Okay, so there are a series of different groups of these developing eggs all throughout the ovary, and they are surrounded by extra tissue. Okay, so that is what the girl's ovaries are like when she is born. Okay, so the egg nests filled with primary oocytes in the primordial follicles are just going to hang out until she reaches puberty. From then on, every month, this series of events is going to occur. Now, keep in mind that all of these different stages, it actually takes quite a long time. So this takes months, even though we are going to kind of abbreviate it and think about what is happening in each and every menstrual or ovarian cycle. Okay, so every month, a handful of primordial follicles are going to be recruited, and they are going to continue their growth. Okay. At this point, the primary oocyte isn't really going to change. It gets a little bit bigger, but the chromatids are just still kind of hanging out, arrested in prophase 1, just like they have been maybe for 10 years, 12 years, 20, 30, 40 years since she was born until this particular month, this particular cycle. Okay. The oocyte is going to secrete a fluid outside of the cell. Okay, so that's this gray substance right here. It is called the zona pellucida, so the lucid kind of clear zone. Now, this is essentially extracellular matrix, and it also is equipped with what are called ZP3 receptors. These receptors are going to be really important for the fertilization process. So this is actually what the sperm bind to as they are fertilizing the egg. Okay, so that's what's happening with the oocyte and the surrounding extracellular matrix. These follicular cells are also going to start growing. They're going to start dividing. Now, it takes a long time for each of these cells to complete mitosis. In fact, it takes about 10 days to double the granulosa cell, so this is an excruciatingly slow process. But ultimately, what we're going to see here is that single layer of follicular cells is going to grow into many of these cuboidal cells, so kind of block-shaped cells. What this should imply is that there's a lot more space in the cytoplasm to actually have a lot of cellular activity, as opposed to these like flattened cells here. There's really not much going on. It's pretty much just a phospholipid bilayer with a nucleus inside and really not much else. Okay, so these are super active cells. Right, as soon as this change happens, we rename the follicular cells to be called granulosa cells. Okay, at this point, there's give or take, about 2 layers. These granulosa cells express receptors for follicle stimulating hormone, or FSH. And so as we're going to see here in this story, FSH is actually going to be driving the development, driving the growth of this surrounding follicle, which includes the granulosa cells, and also a second type of cell is developing at this point. These are called theca or thecal cells, and as we're going to see, they're going to be really important in kind of protecting the outside of the follicle, kind of forming like structure, and also they are going to be responsive to luteinizing hormone over here. This structure, so the granulosa cells and the superficial thecal cells, this is called a primary follicle. The primary follicle continues to grow in response to FSH. Okay, so we look over here, the secondary follicle, and what we can see here is that the primary oocyte is still relatively unchanged. It might be a little bit bigger, but the DNA is still exactly where it's been for the past possibly decades. Okay. The granulosa cells continue to divide, and so this layer gets thicker and thicker and thicker. The thecal layer is also starting to differentiate. As we can see, there is a theca interna, so this cellular layer that is just adjacent to the granulosa cells, and a theca externa. The theca external is made up of CT or connective tissue. There is a layer of smooth muscle developing, and this is going to be really important in the ovulation process. There are fibroblasts, which are specialized connective tissue cells that are secreting all of these protein fibers, and there are blood vessels. So that's what this little red thing is here. This is a blood vessel. And so that's going to be important in essentially delivering hormones to these cells, right? LH and FSH, the gonadotropin hormones. It's also going to be important in bringing nutrients to the follicle and subsequently to the oocyte. And any of the hormones made by the follicle here can jump into the blood and be distributed throughout the rest of the body. Okay. So that's the theca externa. The theca interna, these purple cells here, is responsible for steroidogenesis. Now look how close the blood vessel is to the theca interna cells. That should imply to us that these theca interna cells are taking cholesterol and other they're taking cholesterol and they're taking hormones from the blood and ultimately making steroids. Okay. Also note that these blood vessels are so far away from the granulosa cells that cholesterol really doesn't get down to them. So essentially anything or any steroids that the granulosa cells make must first be started from cholesterol in the theca cells. And so that leads us to talking about the development of the secondary follicle into a tertiary follicle. Generally the same structure and everything with a few key differences. Now, just a note about my diagram here, the granulosa cells are replicating, and so each one of these little boxes here is a granulosa cell, and that would be going all the way around the primary oocyte. Instead of drawing all these little lines in the tertiary follicle, I'm just drawing pink, implying lots and lots and lots of granulosa cells. Now, the granulosa cells have changed since this was a secondary follicle. Specifically, they have started to secrete a fluid into little pockets in between them. And so what you start to see is these little fluid-filled compartments. Right? And, over time, you're going to get more and more and more of those until ultimately they're all going to fuse together to form one big cavity, which we call the antrum. Right? We're going to get there. The theca interna cells express luteinizing hormone receptors. They are also pretty close to blood vessels, which is what's right here. Luteinizing hormone binds to its receptors on the surface of the theca interna cells, and this ligand binding to receptor is going to trigger cholesterol from the blood to be converted into a weak androgen called androstenedione. Okay? So again, because the blood is so close to these thecal cells, cholesterol can easily pass into these cells, and LH turns on the expression of different genes in this steroidogenic pathway, converting cholesterol into androstenedione. And so this is an androgen. This androgen is going to be further converted by these granulosa cells into estradiol or estrogen. In order to do so, these granulosa cells must first bind to FSH, and FSH is essentially going to upregulate aromatase activity. Aromatase activity is responsible for converting the androgen into an estrogen. And so estrogen is going to flow into the blood and therefore be distributed throughout the body. Okay? So ultimately getting estradiol requires both LH to produce androstenedione precursors and FSH to convert the androgen into the final estrogenic content. Okay? Also, we see a little bit of a difference here in the primary oocyte. What I have drawn is cytoskeleton fibers or spindle fibers that are attached here at the centromere. Okay, so where these two sister chromatids are joined together. This is essentially preparing for the completion of meiosis 1. Okay, now keep in mind that all of these different steps have been happening in not just one follicle, but multiple follicles. Right? Again, for each month, for each cycle in an adult woman's life, after puberty, a handful of these follicles, or these follicles primordial, are selected or recruited. They continue development, okay? And finally, right before ovulation, there's the selection of one of these follicles to finally complete the entire process. Okay? One follicle therefore becomes dominant, and it's going to be ovulated, and all of the other follicles that have just developed in this way are going to undergo the process of atresia. So essentially the apoptosis of the cells of the follicle as well as the primary oocyte. Okay, so from here on out, we're going to follow what's happening to the dominant follicle only. Okay. As I said before, in the secondary to tertiary follicle, there are these little pockets of fluid that are forming in between the granulosa cells. By the time the cell or the follicle is mature, all of those little pockets are going to fuse together to form one big fluid-filled cavity called the antrum. That is why sometimes this follicle is called the antral follicle. Another name, and possibly more common name, is a Graafian follicle. Okay, so what that's going to do is it's going to separate most of the spherical follicle from the oocyte and another layer of surrounding Graafian cells. Now these granulosa cells. These granulosa cells that are surrounding the oocyte are called the corona radiata. It's a crown that radiates around the oocyte. Right? It is secured to the overall sphere of the follicle by this little stalk. And again, all of this pink out here are more granulosa cells. Okay. We can also see that there are theca interna cells as well as the connective tissue kind of like sheath all the way around the outside of the follicle. Okay, so that's going all the way around the structure. We can also see is that the process of meiosis 1, right, which was kind of revving up to be completed over here, is now complete. Essentially, one of these pairs of sister chromatids was pulled into one cell, and the other was pulled into another daughter cell. Right? So this one here is this. Okay. Still, we have a copy of chromosome 2, which might have originally come from dad, right? And another one of that same thing. Keep in mind that crossing over has already happened, so there's a little bit of mixing of DNA, but overall, still, this is a copy of this, so a pair of sister chromatids. Okay, so this pair ends up in this cell, which we now call a secondary oocyte because meiosis 1 is complete. The other pair of sister chromatids is pulled into something called the first polar body. Okay, this essentially is genetically a daughter cell, but as we can see, it's really tiny. It's kind of shoved off to the side of the follicle, and we don't really use this for anything. Okay, it's just there to hold on to this extra DNA. We don't need anything from it. Instead, all of the cytoplasm, all of the everything that would have gone into this cell has been absorbed by this secondary oocyte daughter cell. And so in this way, the woman is investing all of her meiotic resources, all of her resources of potentially 4 offspring into one single cell. Okay, so DNA is stored in the first polar body, but it's just kind of garbage. We're going to get rid of that eventually. Okay, so now this follicle is ready to be ovulated. Okay, so ovulation is going to occur when there is a surge of luteinizing hormone in particular, right? Particularly luteinizing hormone. Okay, so this follicle is ultimately going to swell, it's going to burst, and this entire structure in here is going to be ejected out ultimately towards the uterus and fallopian tubes. And so that's what this is right here. So this is what's ovulated. We can see the secondary oocyte. It begins the process of meiosis 2 and stops in metaphase 2. So still these two sister chromatids are joined together at their centromeres. Around that is the zona pellucida, complete with receptors for sperm, if sperm are present. And finally, the corona radiata, which are pretty much just granulosa cells from the follicle. Okay, this ovulated egg will hopefully make its way into the uterine or fallopian tubes, and it's going to be pushed down towards the uterus here by cilia lining the uterine tube. Okay. While this is happening, so while the egg is being pushed towards the uterus and maybe being fertilized, maybe implanting, for these next days, the remaining parts of the follicle here, okay, so all of these thecal cells and all of these granulosa cells remain behind in the ovary, but they don't just die off. In fact, they become even more active. Here, what we can see is that the granulosa cells have grown and become what we call large luteal cells. Okay. The theca cells are going to become small luteal cells. Okay. And something that's really important is that this entire structure becomes even more vascularized. So instead of all the blood vessels just staying out here by the theca, instead they are going to penetrate into the rest of this structure here, vascularizing the entire thing. Okay. What that does is that cholesterol can now be delivered directly to what used to be the granulosa cells, and therefore cholesterol can be converted fairly rapidly into progesterone by these pink, formerly granulosa cells here. Okay, progesterone then can jump right into the blood and be distributed throughout the body. Okay. We still have a lot of the theca cells making androstenedione and therefore granulosa or formerly granulosa cells making estrogen as well. But progesterone really is the most abundant hormone during this phase of the cycle. Okay, this entire structure, which is secreting a lot of progesterone and also estrogen, is called the corpus luteum. Okay, and it is essentially going to maintain the endometrium or the endometrial lining with the secretion of progesterone for about the next 10 days. If this ovulated egg encountered sperm in the fallopian tube, and those sperm were able to pass through the corona radiata, digest a little tunnel through the zona pellucida, and finally bind to receptors on the surface of the egg and deposit their own DNA within the egg, what's going to happen is that meiosis II is going to be complete. This oocyte is going to be a true ovum or a true egg, and it will be fertilized. So essentially, it can begin the process of developing a new offspring or into a new offspring. Okay, and so that's happening as this structure is being pushed along down to the uterus. Now, by the time it gets to the uterus, it's going to start implanting, and so this is about day 6, 7, 8 or so after ovulation, and it is going to undergo the process of cleavage or a modified type of mitosis. Some of the cells of this structure are going to start secreting a hormone called human chorionic gonadotropin, or HCG. When this hormone is released, it can bind to receptors on the corpus luteum, and using a cyclic AMP second messenger system, tell the corpus luteum to maintain itself. Okay, so to keep on secreting progesterone, keep on secreting estrogen, don't die off, we still need you. Okay, and remember that progesterone is what's important in maintaining the endometrial lining. So if there's no progesterone, there's no endometrium, and therefore the fertilized egg is going to be sloughed off with the menstrual period. Okay, so if there is a fertilized egg that starts to secrete HCG or human chorionic gonadotropin, the corpus luteum is going to be maintained. If the egg is not ovulated, it will never complete meiosis II. It will never secrete human chorionic gonadotropin, and therefore, at about 10 days after ovulation, this entire structure is going to start dying off, and it's going to become, or we're going to call it, the corpus albicans, or essentially like a scar of this here. Okay, so again, the corpus luteum is going to be maintained if and only if there is a fertilized egg that embeds within the endometrium, okay, of the uterus here. Okay, and what that's going to do is HCG triggers the corpus luteum to continue secreting progesterone and estrogen to maintain the endometrium until the placenta develops and can start secreting its own hormones. Once again, this has been the process of oogenesis and the subsequent or concurrent follicular growth. Oogonia replicate within a fetal girl's, or I guess embryonic girl's ovaries. The resulting cells are going to begin the process of meiosis I, stopping in prophase I, and just kind of hanging out in little egg nests, each surrounded by a primordial follicle, until they are recruited at some later time, okay, so sometime after puberty. When that happens, the recruited follicles are going to continue to develop more and more cells. In the process, they are going to be secreting estrogen. They are going to be nourishing the developing oocyte. They are going to be creating this antrum. Finally, right before ovulation, the surge in luteinizing hormone is going to trigger this final Graafian or antral follicle to burst, ejecting the secondary oocyte, right, which has begun the process of meiosis II, as well as the corona radiata and zona pellucida. And that's going to go out into the fallopian tubes and down towards the uterus. The remaining follicle becomes the corpus luteum, and it's going to secrete lots of progesterone and, to a lesser extent, estrogen. If the ovulated egg is fertilized Along its journey to the uterus, it is going to develop a chorion, which is going to secrete human chorionic gonadotropin. HCG binds to the corpus luteum and essentially maintains it until the placenta is able to take over this process of progesterone and estrogen synthesis. Thank you so much. Once again, this is oogenesis.